Good evening, everyone, and welcome to our first Genomics Lite session for 2022. Our topic this evening is the human microbiome, and our guest speaker this evening is Hilary Brown from the Wellcome Sanger Institute. Before we kick off with our talk this evening, I'm just going to go through some of the sort of boring housekeeping stuff and then pass over to Hilary. So before we start, all of the events that we host here are done to help us all explore what genomics means for our lives and how it links to our education and curriculum. So we do encourage you to ask questions this evening, but we invite you to do so in a spirit of curiosity and ask you to be respectful of everybody in this space this evening. Myself and Fran are here this evening to moderate the platform to make sure it's a safe space for everybody. In case you've not used Zoom webinar before and not joined us before, you obviously can't use your cameras or microphones this evening, but if you need to get in touch with us or want to ask any questions, you can do so using some of the buttons on Zoom. So you can say hello and answer any of Hilary's questions that he might ask to you using the chat box, and you can ask questions to him using the Q&A button at the bottom as well. We've also got closed captions this evening, so if they're useful to you, you can press the CC button and they should jump up on your screen. You can also move them around your screen, so if they're blocking anything on the screen, you can move them around and hopefully out of the way too much. We're joining you this evening, theoretically at least, from the Wellcome Genome campus just south of Cambridge, which is the home to the Wellcome Sanger Institute and to EBI. Obviously, this is called Genomics Lite, so we're looking at how genomics is used in different areas of science. So it's good to start with thinking about what a genome is. So for what we're doing this evening, a genome is an organism's complete set of genetics instructions and has all the information that an organism needs to grow and develop. So that's sort of the base of what a genome is. And Hilary's going to talk more about how we use genomes and genome sequencing to understand the human microbiome. So the sort of rundown of this evening's talk, I will very shortly pass over to Hilary, who will give about a 30-minute talk on the human microbiome. There'll then be time for a Q&A with Hilary about that topic and about the research that he's going to share with you. Hilary will then talk a little bit longer about his career to give you an idea of how scientists sort of get to where they are, and we'll have time as well for a career Q&A, so any questions about how Hilary chose to go into this line of work, we can answer then. We should wrap up by about 5:45 this evening, and the session will be recorded and then made available on YouTube, so if you need to duck out early and miss any of it, you can catch up afterwards. That is all from me, so I will stop sharing my screen. I will pass over to Hilary for our talk.
Thanks, Sam. Can you see my slides?
Yep, they look great. Okay,
great. Okay, I'll start. Hello, everybody, and thank you for joining us this evening. So my name's Hilary. And as I'm said, I work in the Wellcome Sanger Institute, and I'm going to talk to you today about the microbiome and genomics and how we use genomics to study the microbiome. Okay, so to start with, let's start with a poll. So how many people have heard of the microbiome?
So hopefully on your screen, you'll get a little pop-up that's going to ask you how much you think you know about the microbiome. Oh, quite a mixed result so far. No one has said that they know a lot about it. Lots of people know a little bit or have heard of it but don't know much, and a few of people have never really heard it at all. So a little bit of a mix in the audience this evening.
Okay, good. And so next then is a question. So what comes to mind when you hear the word microbiome?
If you've got any ideas, pop them in the chat box and I'll read them out. Anything at all that comes to mind. Someone's put the gut, 2 people have put the gut, someone's put the digestive system, skin, gut bacteria, someone's put the way that microbes interact in the body, lots of gut bacteria coming up, microbes, more guts, the lungs. So yeah, lots of guts and microbes coming up so far.
Great. Okay, so that's a good start. So okay, so just trying to get, sorry, I'm just trying to get rid of the panel across the top of my screen.
Just be able to drag it sometimes.
Because it's hiding all the slide titles. Okay, never mind, I'll press on. Okay, so I suppose to answer those questions, I'm going to start with another question, and that is, you know, what's in your poo? So this is probably something most of you don't think about every day, but it's something that I deal with quite a lot. So to answer that question, most of what's in your poo is water, 75%, and the rest is solid material. And in that solid material, we have lots of microbial cells. So just to give you an idea of the number of cells, and on your average visit to the bathroom, there is 10 to the power of 11 bacteria. So this is 100 billion bacteria. And it's not just bacteria, there's all other microorganisms as well, such as viruses, archaea, fungi. And then there is some of your own intestinal cells as well. So these are vast, vast numbers that are present in your poos. So, well, why should we care about that? Well, for me, this is how we study the intestinal microbiome. So just to give you a definition, so the microbiota, it's a collection of microorganisms, so bacteria, viruses that live in a particular environment. So it doesn't necessarily need to be the human microbiota. There's microbiotas in soil, in water, in all different environments, in the oceans as well. But today we're going to talk about the human microbiota. And another definition I'm going to use a lot is the microbiome. So the technical definition for a microbiome is the genomes of the microbiota, so the genomes of all the microorganisms in that environment. But people use those terms interchangeably, and you're probably going to catch me using them interchangeably as well. It doesn't really make a big difference. Now, if we think about the human microbiota, so we are all colonized by a dense community of microorganisms that live on us and in us. So you have a microbiota on your skin, in your mouth, in the oral microbiota. There's even a microbiota in your urine, all different parts of your body. But we're going to focus on the gut. So as you move down from the mouth down through the gastrointestinal tract, you get different abundances of bacteria. And these abundances increase until you reach the large intestine, or the colon. And this is where the greatest number of bacteria is found. And even within the colon, you get different communities. So some bacteria like to be attached to the walls of the colon, and some prefer to be floating in the middle, in what's called the lumen. So even within different environments in your body, you get different communities as well. So the point I want to make here is that you're a complex ecosystem colonized by hundreds of different bacterial species. And a good way to think of it is similar to a rainforest, where you have different organisms all interacting with each other, and your body is the same as well. And just to give you an idea of the diversity, so you have different bacterial species on your elbow compared to in the crook of your arm, because some bacteria prefer the dry environment that's on your elbow, and some prefer the more oily environment that's on your skin. Now, these bacteria that colonize us, they're not transient. They're not travelers that are just passing through. We in different human populations, we find the same populations. We find the same types of gut bacteria colonizing individuals. So this picture here I'm showing you, it's a phylogenetic tree, and it's based on comparing DNA sequences between different organisms. So by doing this, we can see how related they are to each other. So all the different colors represent different types of organisms, and the closer they are to each other, the more related they are to each other. So you can see down at the bottom where there's Homo sapiens, that's us and all the other eukaryotes, all the other animals. But really, these are not that diverse. The greatest diversity in the tree of life comes from bacteria. And I've highlighted 4 different groups of bacteria that live within our guts. These are the proteobacteria, the Bacteroidetes, the actinobacteria, and the firmicutes. So these are just different names to group types of bacteria together. And even within these groups, not all of these live in the gut, it's just specific species. So these bacteria, they're highly adapted to living in your gut, and they've evolved with humans as well over millions of years. So for the majority of these bacteria, the only place you will find them is in the human gut, because that's where they are adapted to live. And just to give you some examples of how evolved they are to living in our guts and how adapted they are to the gut environment. So gut bacteria can metabolize carbohydrates, which we cannot. So this means they can eat sugars, which we can't. So a lot of the fiber that we eat, humans don't encode the enzymes to digest these, but bacteria can. So a lot of the fiber you eat ends up in your gut. You can't digest it, but the bacteria that are there love it. And they can break it down. This creates energy for themselves, but it also releases energy. It also releases breakdown products, which creates energy for us, which our body can digest. So they provide, through their digestion, they provide calories to us. Another great example is breast milk. So breast milk contains sugars that are there to promote growth of specific bacteria and not the baby. Now, this is pretty crazy if you think about it, because it means that we as mammals have evolved to produce milk that contains sugars, but the babies can't digest the sugars. The sugars are there to help bacteria in the baby's gut grow, and not just any bacteria, a special type of bacteria called bifidobacteria. So these bacteria, so this is, I suppose, another example of how you can think of yourself as an ecosystem, with you as the human and your microbiome. Okay, so we'll have another poll now, and now I want to ask you what influences your intestinal microbiota. So there's different options. So there's birth mode, where you're born by vaginal or cesarean section delivery, diet, medication, exercise, antibiotics, age, or all of the above.
Awesome, we've got quite a lot of responses in so far. A few people going for diet, but most people going straight for all of the above. So yeah, about 90% of people have gone straight for all of the above.
Great. Because that's the correct answer. So all of these influence your intestinal microbiota. So I want to give you an idea now of how your microbiome changes over time, and how some of your day-to-day living can affect it. So when you're born, you're essentially sterile, and it's at the point of birth that you are first colonized by your microbiome. You first acquire a microbiome. And so whether you're born via vaginal delivery or via cesarean section, you acquire a different group of microbes. And so there's a lot of interest in understanding how this can impact health in later life. So for example, while cesarean section is a very important medical intervention and life-saving, we know that in terms of your microbiome, it's linked to a different microbial community that you acquire. So people are trying to understand what could be the health effects of that later on in life. And then as a baby, whether you're breastfed or formula fed, you will acquire different groups of bacteria. So all the circles here above the individuals, the colors are meant to represent different bacterial groups. And then the medications and the antibiotics you take will have a big impact on your microbiome. I'm going to talk more about antibiotics and the effect that they have in a while. And then generally how healthy you are. But as you age, the composition changes, and the composition you have once you're elderly is quite different from the composition you have when you're a baby or a toddler, or even an adult. And a good way to think of this and picture this is to come back to this idea again of an ecosystem. And so your gut microbiome, it matures in a similar way to other ecosystems. So a good way to think of it could be to think about in a forest. So you have different successions. So if you imagine you have a bare piece of rock that's been recently cleared. So the first things that will grow in there will be quick growing hardy plants, the pioneer species. But over time, these are generally replaced by intermediate species that can grow a bit taller and that are a bit shade intolerant, like pines. So if you take a pioneer species in terms of the gut microbiome, this is when you're born and the bacteria that colonize you in the first weeks of life. The intermediate species could be once you are weaned onto solid food, but then eventually you get a climax community. And so in a forest, this is a very stable community with different trees in it. And in the gut, these are the bacteria that we see in adults that stay with you for the rest of your life. So your gut ecosystem, it matures in a similar manner to other ecosystems. Okay, so now I want to talk about how we study the microbiome and how impactful genomics has been to study the microbiome. So I have a very simplified workflow here of how it's done. So you start with your microbiome sample. In my case, I'm interested in the gut microbiome. So it's very easy for us because we start with fecal samples, because these are a good representation of what's in your gut. And then from this, we extract the DNA and we sequence it. The vast majority of the DNA in feces is bacterial. And then once we have the DNA sequences, we compare them to a database with known bacterial species. And so this is you can think of it as like playing Snap and matching the same color cards together. And so by doing this, we can identify which particular bacteria are in a microbiome sample. So now I'd like to ask you the question. So what can we learn from a microbiome sample by doing this type of analysis?
If you've got any ideas about what we might find out from this, pop those in the chat box and I will read them out. Someone's put if the sample is healthy. Someone else has put health status. So you have some general sort of health related stuff. Someone's put the number and types of bacteria. So you might work out which specific ones are there. Give it another couple minutes. Someone's put what bacteria are thriving due to environmental factors. So again, sort of what type of bacteria. Someone's put maybe the evolution of bacteria, effective diet, links to obesity perhaps, interdependence of the bacteria, what's lacking, and so you might need to have more of. If the patient's recently had antibiotics, and then more about whether it's healthy or not. So yeah, lots about health and lots about what type of bacteria are present in the gut.
Perfect. Yeah, so that's exactly what we can do. We can tell the types of bacteria in there and we can tell their abundances. And really what we're doing in most studies is we are comparing the microbiome composition between healthy people and people with a particular disease. So this could be people infected with a gastrointestinal pathogen. It could be people with inflammatory bowel disease or irritable bowel syndrome or anything. And in a nutshell, this is how a lot of microbiome studies are done. So you compare the microbiome between healthy and sick people, and what you're doing is you're looking for bacteria that are enriched or depleted in one group. So bacteria that are more abundant in healthy people compared to people with a particular disease, or people with a particular disease, you're looking for bacteria that are more abundant in them compared to healthy people. And what you might do then is you might associate those bacteria either with sickness or with health. And this is really powerful and it's really informative, but it's not the full story. So the image I'm showing you here, it shows all the different illnesses or body parts or human physiology that's been associated with the microbiome. And I'm not going to go into the details because there's too many, but just looking at this, you can get an idea of the diversity of human functions that the microbiome is involved in. But there's an important point here, and that is that just because there's more or less of a particular bacterial species in healthy versus sick people, it doesn't mean it plays a role. So this is the idea of association versus causation. So just because something is associated with a particular disease, it doesn't mean it's the actual cause of it. And I suppose I'd like to make this point because the microbiome is in the news quite a lot, and we know it plays a very important role in human health and disease, but sometimes it's hard to separate the hype from what's real. And so the next big microbiome study that's out there, I would advise, if you want to look into it, I would advise you to ask, how many people did they look at? If they looked at 10 people, it's hard to make big claims on that. If they looked at hundreds of people, that's more powerful. And something else is mice are used a lot to study the human microbiome because they're a good model of what happens in the human gut. And the mice studies are really informative, but they're not humans either. So that's something else to consider the next time you hear about a big microbiome study. Is this something that was conducted in humans or something that was based on mouse experiments? Okay, so we have this problem of association versus causation. So how do we get around it? Well, one way to get around it is to do experiments with the bacteria in the lab. So if you do your microbiome sequencing and you identify bacteria that are associated with health and disease, if you have these bacteria, you can do experiments with them to understand, are they actually causing disease, or are they associated with health? So an example could be if you see a group of sick people have high levels of abundance of a particular bacterial species. If you have that species in the lab, you can do experiments to see, does it produce toxins that can make people ill, for example? And this is a big focus of the work that we do in our lab, and that's culturing gut bacteria. So growing them and then using them for experiments. So to do this, we start with our microbiome sample again, which is fecal samples. And then we place the fecal samples on agar that contains different nutrients that bacteria like to grow in. And then once we have colonies, so groups of bacteria growing on the plate, we pick them and then we sequence their genomes. And we also freeze down these bacteria and put them in freezers at -80. And then we can go back to the freezers and grow out the bacteria and use them to do different experiments. Okay, so a lot of the work we do goes on in these anaerobic cabinets. So now I'd like to ask you, based on this poll, so what do you think, why do we use anaerobic cabinets when we're working with gut bacteria? So is it to protect ourselves from harm, to keep oxygen away for the bacteria, or because they look good?
You'll be pleased to know no one's gone for because they look good.
No,
We're not just here spending science money just to make things look good. Most people have gone for the second option, but we've got a few people going for protecting from harm as well. But most people are going for to keep the oxygen away from the bacteria.
Okay, yeah, that's the correct answer. They do, I suppose they do to a small extent protect us from harm in case there is any pathogenic bacteria in there, but most of the bacteria that we work with are beneficial bacteria. So the main reason is to keep oxygen away from the bacteria. So the vast majority of bacteria that live in your gut are anaerobic. So this means they will die very quickly in seconds or minutes in the presence of oxygen. And this is because your gut is an anaerobic environment. So when we get our fecal samples, we try and get them into these anaerobic cabinets as quickly as possible. Now, my pointer isn't working, so I'm just going to describe how this actually works. So on the ground, you can see these petals. There's a blue and a white one. And what you do is you stick your hands into the sleeves that are hanging down. Now, the sleeves, when you put your hands in the sleeves, they're going to be full of air, which will contain oxygen. So you press the blue pedal, and this sucks all the air out of the sleeves. Then you press the white pedal, and this blows nitrogen into the sleeve. So this isn't blowing around the room. This is all contained within the sleeve. So you do this 3 times, and this gets rid of all the oxygen in the sleeve. And then once you've done that, there are doors which you can open, and then you stick your arms inside the cabinet with the sleeves. And the atmosphere inside the anaerobic cabinet is completely anaerobic, so there's no oxygen in there. And that's how we work with these bacteria on a day-to-day basis. Okay, so using this approach, so growing these bacteria, what we've done is we've created essentially a big library of gut bacteria. And so here on the left, I'm showing you another phylogenetic tree, and this time it's just of some of the bacteria that we have grown. And on the right, you can see this image with the box and these little tubes. So these are called glycerol stock tubes. And we mix the bacteria with glycerol, and then we freeze them at -80. And the reason we add glycerol in is that it doesn't burst the bacterial cells from opening. And then once they're stored at -80, the bacteria will stay stable for years. So we can bring them out, we can bring the tubes out of the freezer, and we can streak them out and bring them back into the anaerobic cabinet, and we can work with them again. Okay, now I'm just going to move on. I want to talk about the role of the microbiome in health and disease. And I'm going to focus on one particular example. So antibiotics have a big impact on the diversity of the microbiota. So all of us have taken antibiotics. All of us have, there's probably many listeners here who wouldn't be alive if it wasn't for antibiotics. They are probably one of the most amazing medical discoveries. But when we take them to kill a particular pathogen or a bacteria that's making us ill, they can also do a lot of collateral damage and kill lots of other bacteria in your gut. So if you look at the picture on the left, so this is like a healthy microbiome. And it's the same analogy as a rainforest. So you've lots of species diversity, you've lots of bacteria competing for nutrients and space in the gut. And it's a very crowded environment, and it's difficult for pathogens to come in and colonize and cause infection. And so as a result of that, there is what we call a high level of colonization resistance. So this means it's difficult for pathogens to come in and colonize the gut and make you sick. Now, taking antibiotics is a good way to think about it in terms of what it does to your microbiome. It's like cutting down a rainforest. So you might kill what's making you sick, but you're also going to kill a lot of the good guys as well. And so now we have, looking at the image on the right, you have a reduced species diversity. There's less competition for nutrients and space. And this makes it much easier for pathogens to come in and colonize and cause infection, and you have less colonization resistance. And one particular pathogen that's very good at exploiting this is Clostridium difficile. So this has been, up until, it still is a problem in hospitals, but it's much more contagion and we have a much better understanding of its biology. So Clostridium difficile produces toxins that produce diarrhea. But what it also does is it makes these little dormant structures. You can think of them like seeds called spores. So the image at the top is the vegetative cell. So that's the C. difficile cell that grows and lives inside your gut. But it can also make these spores. So if you look at the image on the bottom right, you can see a spore developing on top. Is the black circle. And on the image on the bottom left, these are the spores. Remember, I told you a lot of bacteria in your gut are anaerobic, and this goes first for Clostridium difficile, or C. diff, as I'm going to call it as well. So this means it cannot survive in oxygen, but the spores can, because they're dormant, and they just, like a seed, they just sit there until you ingest them, and then they germinate, which means they form a new vegetative cell. So the C. diff producing spores has made it a big problem in hospitals because the spores are also very resistant to disinfectants as well. So one of the main, so some people, when they have C. diff, it goes away on its own. Other people are given antibiotics. And this results in about 80% of people are cured. And you can see an image of a healthy colon. This isn't what your colon normally looks like. This is a colon that has been washed, so you can see there's no food in it. But you can see there's like a lovely pink color. But then 20% of people are not cured, and you get like a recurrent infection. And you can see on the right that doesn't look great. So for these people, they're in trouble because the antibiotics are not working. But in recent years, there's been an alternative therapy, and that's the idea of doing a fecal transplant. So it sounds pretty disgusting, but it's extremely effective. And the idea is that you take feces from a healthy person and you give it to the person who has a C. diff infection. And the idea is that the bacteria in the feces from the healthy person will compete with C. diff and drive it out. And it's really effective, to the point where it's available from the NHS as well as a treatment for C. diff infection. And so there's this company based in the States called Biome, and they've made a business model out of this. So they screen healthy donors and then they prepare feces in bottles and they ship them to hospitals all around the States. And you know, it's a great business model. There's lots of starting material. So while this is very effective at treating C. diff, it's not going to be effective for treating all intestinal associated illnesses. So there's been lots of studies using fecal transplants to treat different illnesses, and the figure here highlights some of them. So there's from like C. diff infection, which we know that works, to like IBD, to even HIV and depression and everything. So FMT is not going to be effective at treating all of these. And one of the reasons is it remains undefined. So you don't know exactly what is in the sample. So in a particular fecal sample, there may be some bacterial species that are missing that could treat a particular disease, but if they're not in the fecal sample, then it's not going to work. So one of the approaches that we're doing is, and one of the questions we're asking is, can we identify good bacteria to treat particular diseases, especially pathogen infection? So instead of using a fecal transplant, you are trying to identify a particular bacterial species using our culturing methods and use that instead to treat an infection. And so the commensal beneficial bacteria in your gut, they use lots of different ways to compete with pathogens. And these are some examples here. So I mentioned already the indigestible carbohydrates in fiber. So when you eat these and your gut bacteria produce them, one of the things they produce is called short chain fatty acids. And so these are an energy source for the cells in your gut, but they're also quite inhibitory to a lot of pathogens. The commensal bacteria in your gut, they can also compete directly with pathogens for food, for nutrients, and some of them even produce these antimicrobial peptides, so like bacteriocins, and these are like toxins that are quite specific to kill particular pathogens. So these are just some examples of how the beneficial bacteria in your gut can outcompete and get rid of pathogens. And this is a big part of the work that we're doing in the lab at the moment, is trying to identify bacteria, beneficial bacteria that have this effect. And these could be a treatment for people with a particular pathogen infection instead of trying to use FMT. Okay, so I'm going to finish up there, but I'd like to finish with acknowledging all the people that I work with, based in Sanger and also in EBI on the site we work in. And I've had the privilege of working with a great number of really talented people over the last few years, and these are the people who've contributed to a lot of the work I've talked about today. So thank you very much. Thank you for listening.
Awesome. Thanks so much, Hilary. If anyone's got any questions about anything that Hilary just talked about, pop them either in the Q&A in the chat box, and we'll sort of read some of them out to get answers for. My sort of immediate question, because obviously you said that you work with fecal matter, is there anything that like gets missed out in fecal matter that might be there in the gut that doesn't end up in poo? Like any bacteria that you sort of don't get to see because they stay in the gut?
Oh yeah, sorry. Yeah, that's an excellent question. So the bacteria in poo are a good representation for what's going on in the colon in the large intestine. And they're not as good for what's going on in the small intestine, although we do know that some bacteria will pass from small to large intestine and will survive, because they're small and large intestine are different environments in terms of the pH in there. So the bacteria that like to live in the small intestine don't like living in the large intestine. Another thing is that some bacteria like to stick very tightly close to your epithelial cells, and so poo might not be a good way to see these if they don't come out. But you have to remember as well that your epithelial cells in your gut, you're constantly shedding these as well. So while it's not perfect, it's quite a good representation of what's in there.
Awesome. We've got a couple questions coming in, so we'll go through these. So firstly, you mentioned that anaerobic conditions are needed to study these bacteria. They've asked, are there any effects on exposing your hands to these conditions?
No. So we wear gloves in the lab. So because of the bacteria that we work with, we wear gloves and lab coats. Because sometimes we do work with pathogenic bacteria that could make you sick. And sometimes, there might be a small chance of something pathogenic being in a fecal sample. So it doesn't, the anaerobic environment doesn't have any effect on your hands, but we're always wearing gloves in the anaerobic environment as well. The main drawback is actually, and especially in summertime, it's quite hot and sweaty working in there, because the anaerobic cabinet, it's anaerobic, but it's also at body temperature at 37 degrees.
I guess that's pretty important to keep it at that, right? You can't like open windows and stuff because that would sort of ruin the environment.
Yes, yeah. So everything inside the cabinet is meant to represent what's in your body. So it's 37 degrees, and there's no oxygen in there.
Someone's asked, why do older people lose a healthy microbiome, and how can they help maintain one?
Yes, a great question. So yeah, as you get older, your microbiome becomes less diverse. So some of the species that were there when you were younger disappear. It's probably due to a changing immune system as you get older, and it's also probably due to a changing diet. People eating a more simpler diet as they get older. I suppose some ways to remedy this is to try and ensure you eat lots of fiber and lots of fruit or veg as well. So these will all contain carbohydrates that are very good at nourishing the bacteria in your gut.
Sorry, I muted myself there. Someone had asked, in the graph that you had about the fecal microbial transplants, atopic dermatitis is listed. How is this condition related to the gut microbiome?
Yes, that's a great question. So your gut environment is one of the most immunogenic parts of your body. So your immune system is highly switched on there. And this is because if you think about it, you're ingesting food and you're bringing stuff from the outside into your body. So your immune system has to be highly tuned and quick to pick up anything that shouldn't be there. So there's a lot of research looking into this, and people think that changes in your gut microbiome can affect different allergies like atopic dermatitis. And this is because your immune system is interacting with the bacteria in your gut, not in a bad way. So the commensal bacteria in the gut help educate and train your immune system. But there's a thought that if you're missing certain species, then your immune system can develop in a different trajectory, and this can lead to allergies in later life, or something like atopic dermatitis.
Awesome, thank you, and good spot to everyone whoever spotted that on the graph, because they're quite small bits of text, so well done for spotting it. Someone's asked a nice sort of broad question. What has been the most interesting discovery you have made about gut bacteria during your time at Sanger?
So I have a big interest in spore forming bacteria. So I mentioned C. diff, which produces these spores. These are like bacterial seeds that help it transmit from person to person. But I also found out that a lot of the beneficial bacteria in your gut can also do this as well. Actually, the majority of bacterial species in your gut can make spores. And we think this is because it helps these anaerobic bacteria transmit from one person to another, so they can survive, so the spores can survive in the outside aerobic environment until they're ingested by a new host, and then the spores germinate, forming a new vegetative cell. Because something I didn't mention is that we share our microbiome with each other more than you realize. So the people you live with, you will have a more similar microbiome with them compared to people that you don't live with. And this is because we're sharing bacterial species between us. So that was the most interesting discovery for me, was that a lot of these bacteria can make spores, which help them transmit between people.
Isn't there like a fact about sharing bacteria with your dog being like the highest I'm sure I've seen that.
Yeah,
Dogs
there
and
was a paper from a few years ago where people who had a dog had a more similar microbiome compared to people who didn't have a dog. And the thought was that the dog was helping to transmit the bacteria. But a dog's microbiome will be quite different to ours as well. And I suppose it's just to highlight the point that the bacteria that live in your gut, in the human gut, can be quite different to bacteria that live in the guts of different animals.
awesome. You said during the bit about antibiotics that the microbiome can decrease in diversity. Can it ever be the other way around? So can your biome diversity increase after antibiotics, or is it always decrease?
Can it increase after taking antibiotics? I suppose it can, yes. So if you are infected with a pathogen, so a lot of pathogens can, one of their pathologies is that you produce diarrhea, and so this will reduce the diversity of bacteria in your gut. So taking an antibiotic to get rid of a pathogen will restore the balance in your gut and allow those beneficial gut bacteria to come back as well. So I think that's probably the best example of how the diversity can increase after taking antibiotics, if you're successfully treated for a particular infection.
Awesome. We've had a more amusing question. Is it true that the gut microbiome can influence your food cravings?
I don't know.
Or is this one of those, like you saying earlier, that there can often be a lot of hype around the microbiome? So I wonder if that's a case of that rather than of anything else.
I think it probably is. I don't know. I never really thought about that. There's a lot of research looking into the role of the microbiome in obesity. There was this is a few years ago now, and there was a lot of research looking into it and suggesting that the microbiome did play a role in obesity, but I don't think we're fully there yet in understanding if that's actually the case. So I suppose you could build on that and say there might be an argument then for if you have certain bacteria that metabolize a lot of the food in your gut, then you might get more hungry.
And then sort of last one we'll do for now before you can talk a bit about your career. Someone's asked, what do you think the future of the biome transplant technology is, the sort of fecal transplant? What do you think the future of that is?
I think it's really exciting. So there's lots of companies have started using the microbiome as a therapeutic. So I think the future will be identifying particular bacterial species that can treat a particular disease. And then you know exactly what you're putting into a person. And then it becomes much more of a defined medicine or a drug. And I think that probably some of the exciting areas are in treating pathogen infection, but there's also a lot of research now into cancer treatments. So there's a particular cancer treatment called checkpoint inhibitors, which are designed to provoke your immune system into attacking tumors. And they found that some people are better at this, and they found an association with the microbiome. So there's this idea that if you could give people particular bacterial species, these would interact with the immune system in a certain way, which would then trigger the immune system to kill tumors. So I think this is one of the really exciting areas, and not quite doesn't seem like quite an obvious one when you think about gut bacteria.
Sorry, I was on mute, wasn't I?
Yeah,
always a good start. There's a couple more questions that we'll put a pause on for just now, and I'll let you talk a little bit about your career, because there have been some career questions coming in, and then we'll do some more questions about your career or any other questions that come in.
Okay. So I'm going to give you a background to my career and how I ended up here, and any thoughts and observations are just from my experience. They might necessarily be right. So I'm from Cork in the bottom of Ireland, and so in terms of my education and my career, I started by doing a degree in agricultural science in University College Dublin, and this was in part because I was interested in science, but also because I grew up on a farm. So doing agricultural science seemed like a good way to mix that. And then after that, I came back to Cork and I did a master's in biotechnology. And this is in University College Cork. And it was while I was doing that master's, there was one particular course, and it was on genetic engineering. And this kind of started my interest in genomics. And then when I finished the master's, I didn't really know what I wanted to do, and I hadn't really done a lot of traveling at that point. So I applied to this Irish graduate program. So this was for Irish people to go and work in Japan in companies. So these companies had factories or bases in Ireland, and the idea was you go to Japan for 2 years and learn the language and the culture and work in the environment, and then some of the people might come back to Ireland and work in the companies. So I spent 2 years working there in a company in Kobe down in the south of Japan, and it was a fantastic experience. So the work was this company that made cleaning solutions for contact lenses, which was something I knew I wasn't interested in making a career out of, but it was an opportunity to go and work in a completely different environment, in a completely different culture, and it was a fantastic experience. And then I finished that, and my girlfriend at the time, my wife now, was just about to move to Sydney for a year or two because her sister lived there. So I followed her then to Sydney, and I ended up spending a year in Sydney, and I spent a large part of that working in a bank in like a project management role. And again, this was something that I knew I didn't really want to make a career out of, but it was an opportunity to do something different. And then my visa was expiring, and I had to leave Australia, and I was scrambling around looking for jobs, and I was lucky enough to get a job at the Sanger Institute. And I'd never heard of it before I got the job. But I took the job, and it was as a computational biologist. So this was doing a lot of manual work, trying to improve bacterial genomes. And then after a few years doing this, I switched to a research assistant role. This is in the lab I'm in now. And then I enrolled in a PhD program in the London School of Hygiene and Tropical Medicine. And then when I finished my PhD, I became a staff scientist at Sanger. So a staff scientist it's kind of a central role within research teams. So you'd have your own research projects, but you're also involved in helping other people with their research projects as well. So over the years at Sanger, I've kind of progressed and moved from one role to another. And I mean, my take-home messages from this would be, if you enjoy something, do it. I was interested in genomics. So when I had this opportunity to work at Sanger, even though I didn't know anything about I'd never heard of it, and I didn't know anything about the role, I thought it was a great opportunity to work in an environment that studies genomics. I suppose something else is, from my experiences, you don't need to know exactly what you want to do. I didn't have a clue what I wanted to do when I was in my 20s. So when I was leaving Ireland for Japan, or even in Australia, I was still trying to figure out what I was going to do as a career. And I think that's fine. You don't need to rush. I think if you are interested in a career in biology, and especially in academia in biology, I always think that the why is more interesting than the how. So the how is interesting as well, but the why is more important. So why do pathogenic bacteria produce diarrhea? And the reason is, of course, is they're not doing it to make us sick because they don't like it. It's because it's easier for them to transmit from one person to another. I think having different skills, so for example, lab skills or computational skills opens doors. So if you have some lab experience, and if you're interested in coding as well, this is quite powerful, because you can work across different technologies. I think if you have the opportunity, you should travel and see the world. I've found it to be an amazing experience that has definitely improved me as a person as well. And take your opportunities. So I had the opportunity to go and work in Japan, and I took it. I didn't really know much about it, but I went for it. And it was the same when I moved to Sanger as well. They don't come around that often. It doesn't have to be perfect, but I think if it sounds like something that you think you could like, I'd give it a go. And then the final thing is in terms of looking for career advice or work placements or secondments, don't be afraid to reach out to people and email them. If you do, try and make it a bit personal. So, for example, if you're looking for a placement in a lab somewhere, say, I'm really interested in your work because of X, Y, and Z, or I read a paper that you recently produced, or I saw you doing an interview on whatever, and I'd like to learn more about it. But don't take it personally if you don't succeed or hear back. A lot of people are very, very busy, and not everybody has the time to respond to emails. So it's not a reflection on you. It's just a reflection on the time that people have. And that's it, really.
Awesome. Thanks so much. It's always just nice to hear how different people got to where they are. And I think you've had a really interesting career and clearly, yeah, got some really nice traveling as well. So if anyone's got any questions specifically on Hilary's career, stick those in the Q&A or the chat box. I always like to ask our speakers what your sort of favorite bit of your job is and maybe what your least favorite bit of your sort of current job is.
That's a really good question. The favorite bit is doing the science, the day-to-day stuff. The least favorite bit is the admin. That goes with it. But that's probably the same for any job.
I currently can't see any questions specifically about your career, but there was a good few that came in sort of about your main topic. And one that I think is really good is someone's asked if they want to read more about this topic, how could they find useful websites or sources that don't have the hype that you were talking about? So that it's sort of just the core science rather than any of the hype.
There's lots of really good books out there. Maybe I can, I don't know, is there some way to share them afterwards?
Yep, so if you send them to me, I can pop that out on the Eventbrite email list so that everyone has them.
So there is some good popular science books that are quite informative and very educational as well.
Awesome. Yeah, so keep an eye out for that, and I'll pop that around when you have a chance to send it through. Someone's also asked, back on the sort of science, are there any longitudinal studies that have followed people over time and looked at their microbiome and what happens to them in terms of disease and health? I'm sure there's lots, but does any specific come to mind? Yeah,
there's lots, and I think this is where a lot of microbiome studies are going. So, for example, we're involved in a project looking at babies born either via cesarean section or via vaginal delivery, and so we follow them for the first few months of life. But what we'd like to do is follow them for the first few years of life, because this is when kids start to develop like asthmas and allergies. And so if you can imagine, if you have hundreds of kids and you have their health records and you know what medications they've taken and how they were born, and even if you can get like information from parents on whether they're breastfed or formula fed, for example, if you can follow those kids for years and sample their microbiome, you're going to build up this amazing picture of how their microbiome is developing. And then by using the medical records and looking for differences in the medical records in the kids, I think we will start to link these events in early life to health in later on. So we know that kids born via cesarean section have higher rates of asthma than kids who are vaginally delivered, and the microbiome has been shown to play a role with that. But what we want to do is to really follow these kids for longer and to have larger numbers as well. And this makes the statistics behind it much more powerful to link the microbiome to these illnesses.
Awesome. Someone's asked, how has research into the microbiome influenced medicines used in sort of probiotics and prebiotics?
So traditionally, a lot of the probiotics that are available in shops are lactobacilli or bifidobacterium. And in adults anyway, these bacteria are not abundant in the gut. So bifidobacterium is very important in the infant gut, but the bifidobacterium species are less abundant in adult guts. And similarly with lactobacilli, it's not a big player in terms of abundance in the gut microbiome. So there's this big debate about what effect these probiotics are actually having. So some believe it's the effect they have is as they pass, so they cannot colonize in the gut, but the effect is as they pass through the gut and interact with the immune system or interact with different bacteria as well. I think the probiotics we will see in the years to come are going to be quite different from probiotics on the shelves now. They're going to be based on a lot of the research that's going on now, and it could be quite different. I think the prebiotics is also something that's really interesting, because we know that different bacteria prefer different carbohydrates and different sugars, and so you can target particular bacteria by giving a particular prebiotic. You can help them to grow in the gut, and there's been a lot of research into this and to stimulating the growth of certain bacteria, for example, in kids who are malnourished, for example. So yeah, so the probiotics is a really interesting one in the gut microbiome. It always gets a big argument going, but I think they will change in the future.
Awesome. I think we've just got one or two more at the moment. So someone's asked, can an unhealthy, or I guess sort of lack of diversity in the gut microbiome influence mental health conditions? This is definitely something I think that's been in the news a bit, and I wonder if it falls in the hype or what sort of science there is behind it.
There's a great interest at the moment in the gut brain axis and how the microbiome in the gut influences the brain. So for example, through metabolites that are produced, that are picked up. I think it's really, really interesting, but I think it's at a really early stage yet of development. Most of the studies behind it have been carried out in mouse models, which are really informative, but they're not humans. So I wouldn't say it's hype. I would say it's just too early to tell what effect it actually has, or if there's something that can be actually developed into therapeutics as well. I think it's early days.
Awesome. And I think by the looks of it, we've got through all of the questions that came in this evening. So if anyone's got any last questions, do pop those in the chat box. But if not, I'll thank Hilary so much for joining us. I've definitely learned quite a lot about the microbiome this evening. I don't think it's a topic that gets talked about enough. So yeah, very, very interesting for me as well. And thank you for answering all the many questions that were great that came in this evening. I will just share my screen to wrap us up. So we've got another Genomics Lite happening next month, and our topic then will be evolution and proteins. We've got a speaker, Zachary Arden, who'll be joining us to talk about how proteins evolve and how new proteins can be sort of brought about through evolution. So the link for that I'll pop in the chat box in just a second, and it'll be in your Eventbrite catch-up email tomorrow as well. Otherwise, thank you so much for joining us. If you've got any sort of questions after you've left, you can always email us, and I'll pop our email in the chat box in just a second as well. But yeah, thank you very much, Hilary, for joining us, and thank you for everyone for tuning in this evening.
Thank you very much.